Hi everyone, my name is Bian Li. I'm a postdoctoral fellow at Vanderbilt University. First, I would like to thank the organizers for this opportunity to present my research here at ISMB. In the next couple of minutes, I will be talking about a framework that we developed to quantify the within-human species constraint on amino acid residues in the context of three-dimensional protein structures. Our work was partly motivated by the open problem of annotation of variant pathogenicity. Large-scale genome and exome sequencing efforts in the past several years have uncovered millions of missense variants in humans, and many of these variants are believed to be playing important roles in human health and disease. Unfortunately, the pathogenicity is known for only a tiny fraction of these variants. While several high-throughput functional assays were developed in recent years for several genes, experimental characterization of all variants is still impractical due to the time, cost, and labor intensiveness of these high-throughput functional assays. To help address this challenge, many computational tools have been developed since the early 2000s. Computational tools are attractive because they are often highly efficient. However, these in silico tools are still not sufficiently accurate for clinical variant interpretation. Because of this, the development of novel, more accurate tools is still an active area of research. Nevertheless, one important common feature among these tools is that most of them take evolutionary constraint into account one way or another. One particularly interesting group of methods developed in recent years are what we call population genetics-based constraint metrics. We say that a genomic site or region is constrained if the observed amount of genetic variation in it is less than expected. To illustrate this, let's think of the landscape of genetic variation in the current human population as a map of bullet holes in aircrafts returning from battlefields. Some parts of the aircraft are devoid of bullet holes because they are likely functionally important. When the aircraft got hit in these parts, it is unlikely to make it to return. Similarly, regions of the genome devoid of mutations are likely to be under constraint. When the genome gets hit by mutations in these regions, the genome is unlikely to survive selection. There have been several metrics that implemented this idea. Roughly speaking, all these methods scan one-dimensional sequences for regions that have less variation than expected. As shown inside the papers cited down here, these metrics performed well in various variant interpretation and disease gene discovery tasks. However, the important aspect that we think was missing from these existing approaches is that they do not account for the functional context of the protein coding variation, the three-dimensional structure, and quantifying the population genetic constraint in the context of three-dimensional structure is our focus. Here's what we mean by 3D structural context. We all know that amino acid residues form 3D contexts when 1D sequences fold into their native 3D structures, and that neighbors in 3D space are not necessarily neighbors in 1D sequence. In fact, long-range 3D contexts that are more than 15 residues apart in sequence are very common. Our survey of structures in the protein databank and high-quality models in the Swiss model repository indicate that, on average, about 30% of the contacts each residue makes are long-range. In our approach, we hypothesize that amino acid residues are constrained by spatial interactions so as to maintain the protein's structural and functional integrity. Thus, we expect that consideration of genetic variability in 3D spatial neighborhood should give a better quantification of constraint. we define the 3D neighborhood of the residue of interest as the set of residues that are within 8 angstroms, and we call it a contact set. To implement these ideas, we developed a framework called the Contact Set Missense Tolerance Score, or COSMIS. Here is how the framework works. In a nutshell, our framework estimates the constraint on a residue of interest as the depletion of missense variance in its contact set compared to the number expected if it were evolving neutrally. To compute the COSMIS score of the residue of interest, we count the number of unique nomad missense variants in its contact set and compare it with respect to the distribution of expected number of missense variants simulated based on sequence context-dependent missense mutabilities, and we compute the z-score of the observed number of missense variants and designate the z-score as the COSMIS score. According to our definition of constraint, a residue with a lower COSMIS score indicates that there is a greater depletion of missense variants in its 3D spatial neighborhood, and hence the residue is more constrained. Considering both structures from the protein databank and high-quality models from the Swiss model repository, we were able to compute the COSMIS scores for over 2 million unique residues. Here is what the overall distribution of the score looks like. As expected, the score spans a wide range from negative to positive with a clear shift toward constraint. For the rest of the talk, we define a residue as a constraint residue if it has a COSMIS score of less than negative 2.33, corresponding to a p-value of less than 0.01. With this framework at our disposal, the first question we asked was, does COSMIS score identify disease-relevant genes? Since the PLI score is one of the most used metrics for determining the disease relevance of genes, we looked at the distribution of COSMIS scores for genes in different PLI categories. We found that on average, genes considered intolerant by PLI has a significantly more constrained average COSMIS score than those considered tolerant. We also looked at the COSMIS score distributions across various gene classes. We found that as genes become more essential, on average, their residues have more constrained COSMIS scores. Overall, our analysis indicates that COSMIS score captures well the disease relevance of genes. One of the strengths of COSMIS is that it quantifies constraint at residue level in 3D structural contexts. As a result, it should provide constraint at a more refined scale than metrics that work at gene or transcript level, and it should also reveal structural insights. One of the examples that illustrate this strength really well is a gene called the ubiquitin-like modifier activating enzyme 5, or UBA5. UBA5 has a 0 PLI score. According to PLI, it is the least possibly intolerant. However, this is not the case according to COSMIS. We computed COSMIS scores for UBA5 residues, and we found that residues involved in UBA5 dimerization and protein-protein interaction are more constrained than the rest of the protein. On the right-hand side, we also highlighted the top 10% most constrained residues according to COSMIS. It turned out that most of them play important functional roles. In fact, it was recently shown that mutations at some of these residues are associated with early onset in encephalopathy. The next question we asked was, how well does COSMIS score predict variant pathogenicity? To answer this question, we computed the COSMIC scores for over 4,500 variants whose pathogenicities have been unambiguously annotated in ClinVar. Our analysis indicates that, on average, residues that give rise to pathogenic variants have a significantly more constrained COSMIC score than residues that give rise to benign variants. We also compared COSMICs to 4 other similarly constructed metrics that did not incorporate 3D structural context, and we found that COSMICs performed substantially better in terms of predicting pathogenicity. Overall, our results suggest that considering 3D structural context boosted performance. I have shown you that COSMICs performed very well in various tasks, but computing the score requires a protein structure. Now you might be wondering how much of the coding genome can we compute the COSMIC score for? It turned out that the 2+ million unique residues that we were able to compute the COSMIC score for represent roughly 50% of the coding genome, despite the fact that we selected structures and models using relatively strict criteria. Specifically, the COSMIC scores for 26% of the coding genome can be computed using structures from the protein data bank, and the COSMIC scores for an additional 21% of the coding genome can be computed using high-quality models from the Swiss model repository. And the framework is certainly not limited by the existing structures and models in the protein data bank and Swiss model repository. It is actually straightforward to expand the coverage of the framework using custom-built homology models. For example, we built homology models and computed COSMIC scores for over 40 ion channels for which no structures or models existed in the protein data bank or Swiss model repository. And in this analysis, the custom-built homology models did not compromise the ability of the COSMIC score to predict variant pathogenicity. In fact, COSMICs achieved excellent area under the ROC curve for predicting the pathogenicity of ion channel missense variants, as shown in this ROC curve. Now let me briefly summarize our work. We developed a new framework called COSMICs for quantification of the constraint on within-species genetic variation in 3D neighborhood. We computed the COSMIC scores for over 2 million residues, covering about 50% of all canonical transcripts in the human coding genome. We showed that COSMIC score captures well the disease relevance of genes and that it provides biophysical insights into potential functional roles of constraint sites. We also showed that COSMIC is accurate in predicting variant pathogenicity. And we further demonstrated that COSMIC can be used with custom-built structural models without compromising its ability to predict variant pathogenicity. And finally, we made COSMIC publicly available at our GitHub repository. In the near future, we will create a browser for interactive exploration of COSMIC scores in 3D structures. We will also update the scores as NOMAD expands. And to further expand the coverage of the framework, we will build more homology and de novo models. And to account for severity of amino acid substitutions and allele frequencies, we will likely to develop a new formulation of the score and test to see if it will further increase the performance of the score. I will conclude my presentation with a big thank you to my mentor, Dr. Tony Capra, and colleagues in the Capra Lab for the comments and suggestions that have continuously improved this work. I'd also like to thank the financial support from the American Heart Association and the computational support from the Vanderbilt Advanced Computing Center for Research and Education. And thank you all for your attention.